That's how we convinced FDA that whatever put in is safe, and when they hydrolyze, they come out safe. So that's how we got 15 510K approvals. Abrex got 15 510K approvals. Matter of 18 months, they were able to develop a product. Now they have over 10 products for all orthopedic applications.
Please listen to the disclaimer at the end of this podcast.
Hello. Welcome to the pod, everyone. Most of the stuff in healthcare, we put medical device into the body to last. But today's guest, his main inventions are to build things that last in the body but then vanish. From dissolving sutures to printable tissues to even biodegradable plastics. Our guest today, Dr. Ra Bezwada, is the scientist behind a whole slew of products of these bioabsorbable polymers. Welcome to the podcast. So Ra, you're a chemist who made almost 150 inventions in your lifetime, but I know that you've been in the industry for more than 40 years. Is that right?
Right.
So tell us, what was that like you started in the 1990s, some of our audience wasn't even born. Tell us, what was the journey like back then as a material scientist?
Okay. Let me give you a little background. Thank you, Jenny, for the podcast, and thank you for your introduction. I came to the United States after my graduation from India Medical School, and I've joined STEM Institute of Technology, Hoboken, did my master's and PhD in polymer science, actually polyurethane science. And I was fortunate enough to get a job in Ethicon medical device company, the leading medical device company. And the timing was so perfect. I was in 1980s, they started working on absorbed polymers. First time before that, we using the catgut suture, which is a collagen-derived suture material, which is not very good. So they got the assignment of this project for me, and we developed the monocle suture, which is far better than catgut suture, which is 2-3 times stronger, more flexible, good tie down. This is a terminology, tie down strength as terminology for sutures. Surgeons understand that. So we developed it and introduced the product. It's a great success for the company. And it was annual sales is now $100 million. Cumulative sales is $3 billion of the product. It's a successful product. When I was working on this product, I realized there's a lot of opportunities to make all kinds of absorbable polymers, liquid polymers, elastomers for drug delivery. I'll explain more on the other polymer stuff later.
So was Ethicon part of Johnson & Johnson, or it was bought by Johnson & Johnson?
It's a part of Johnson & Johnson. They started as it was not purchased or anything. It was developed in-house. It was a leading 85% market share at the company. It's still a leading company. It's the number one company. It's a great company to work with.
So you decided to study bioabsorbable. Did you have the vision that a lot of things in healthcare wasn't perfect because it's not absorbable by the body at the beginning? How did you decide to focus on this field?
So fortunately, I worked on Ethicon on absorbable polymers, and I realized a lot of potential for absorbable. So the applications are enormous applications. Can you imagine have a polymer which does its job and go away from without doing any harm to the body? So we can save the so many for the polymers. So what I did was that find out what the polymers were developed. Let me explain to you before I go too far with this stuff. Let me give you an example of what is absorber polymer, finish absorb polymer before I go too far with this stuff. And a little bit of history. It's a great history with this stuff. It was discovered, the product was discovered accidentally by DuPont.
I
DuPont
see.
was DuPont was working on fabrics in 60s and 70s, and polyglycolic acid is one of the products. They made the fabric and test for the washing cycles, go through the cycles, and they found out that nothing came out. And then they realized this polymer is not stable under moisture and heat. Then DuPont decided they don't have any use for it, licensed the technology to Ethicon. So Ethicon gained their product. It's a $6 billion business, absorber polymer business. So
Amazing.
amazing. So
Yeah.
so over the years, it was only 50 years history in the whole absorber polymer. Over the years, first they developed a suture material from DuPont chemistry. That was the PLGA polymers, polyglycolic polylactic acids. They developed a monofilament suture for PDS polymer. And I developed a monocle suture after this because the PLGA polymers are too stiff, very high melting. Processing is going to be difficult. Making sutures, especially monofilaments, is going to be difficult. So they asked me to work on most best handling properties of suture stuff. That's why they took monocle. So I discovered this polymer by making a segmented copolymer. First time making a segmented copolymer of polycaprolactone and glycolide. So the polymers we have earlier was a homopolymer and a copolymer. PDS is homopolymer, PLGA is a copolymer, and they're very hard to process them. So when I introduced the monocryl, it's a big hit, and then they replaced the catgut suture. It was mainly used for most gynecological procedures and plastic surgery.
Why is that? Because it's more internal?
Because it is the plastic surgery, which it goes away very quick and also very easy to handle, tie down, because the tie down is very important. Good strength when using it in the earlier stages of the stuff, and go away from absorption cycle, very fast cycles, and also BSR strength goes away. So plastic surgery is easy, very good. Gynecological procedures,
So back then, when you were doing just pure science, and this still fascinates me for the work that chemists do. It seems to be alchemist in a way, like you just decided how to mix this and that together, and suddenly things happen. But what was I don't know if you can tell us, what was the process of discovery, this
Oh,
monocryl-based polymer?
that's an excellent question. So what happened with we are making all kinds of sutures, absorbable sutures, and it came to my mind, what is the cause for the hydrolysis breakdown? Why one polymer hydrolyzes faster than the other? So I looked very carefully, analyzed it, and found out that glycolic acid, the very short chain of the CH2COO, that's a short link of the polyglycolic acid, hydrolyzes very fast in the presence of moisture. I said, and then I reviewed all the commercial sutures materials and found out the only products which are absorbable, fast absorbable, are the ones containing glycolic acid. So I thought, if I can find a way to take this glycolic acid, incorporate it into the backbone of the polymer chain, if we could do it, it will control the rate of hydrolysis of the polymer, thereby by putting the glycolic acid in the chain, which will increase the hydrolysis if we get closer to it, if they are far away from it, also neighboring group effect of the stuff. That started my discovery process of making a novel new absorbable polymers. That was the so from that technology, I developed all these polyurethanes, tissue adhesives, montage, everything, and it's still going on now. I even have bioconjugated drugs we're making it with this stuff.
Absolutely. It's kind of amazing because in software industry, we always hear founders pitch platform technology. And very rarely people would understand that chemistry can be also a platform technology as well. And it seems like you have discovered the beginning of a platform technology
Right.
in the chemistry side, which is kind of amazing because the number of polymers you could modify using this discovery is probably infinite.
Unbelievable. Unbelievable.
So,
It's
so, 20,
go ahead.
so back then when Ethicon was hitting the lottery, got the jackpot, you could have write your career successfully there forever, probably till today in Johnson Johnson and Ethicon. What made you decide that you're going to just give out all that and start your own company, which is not an easy life, I would say?
Oh, I agree with you 100%. I thought came to my mind so many times, why did I do this stuff? One of the main reasons is actually I have nothing but good words about Ethicon and Johnson Company. So when I started working in Ethicon, they have most of the companies have exploratory research group. I don't know whether it may be too young for this stuff. So every DuPont, AT&T, General, all the companies have a 3M is the number one exploratory group. I happen to work on exploratory group, a lot of freedom. We can work anything I want with as long as they get a boss permission. So you're able to do the stuff. So that's why I was able to be very successful. I got an average of 3 patents per year. Every year, there's a history in Ethicon. I think Ethicon, I was the only guy got this so many patents for this stuff. So what happened over the years? Research changed, project management changed, and they put lid on the exploratory research. Actually, eliminated exploratory research work, and the projects are run research run by project leaders. So the direction goes in a different way, very limited flexibility. So I wanted to explore all my technologies what I have developed. So I took a chance to come out and do this stuff. And I think against Ethicon, which was a good company, but I had the opportunity and the freedom to develop more products. That's why my own company have 55 patents because nobody stopped me to do the patent. Um, so even though it cost me money, was able to accomplish a lot of technology with our patents.
So after almost 20 years career at the large medtech company, you decided to quit all that and
Yeah,
start your own company. What was the
right.
first 1 or 2 years was like? And back then, VC was not a
Yeah.
thing. You probably
So
have to be on your
that
own.
actually you are asking me very good questions. What happened is that at the time, stent business was big, booming. I don't know whether you recall that in 2000 to 2005 timeframe, um, stent business was booming, and there was a lot of need for new polymers. And if you make a new polymers for stent coatings, absorbable stents, people are paying, giving money, millions of dollars of research work. By the time I decided to quit and do the stuff, and the stent business had problems, so that money was ran out of the money with the stuff. But fortunately, I contacted one of the polyurethane company guy in California, met him in ACS meeting. He was supportive for 2, 3 years to develop the absorbable polyurethane work. With his funding, I was able to do initial work, which was very helpful to me. Unfortunately, the the person was company sold. He sold the company and left. So I left out with uh, problems, the financial problems with the stuff. But it was not too long. Within a few months, I was able to get funding. So I also do the contract research work, custom synthesis work, absorb polymers while I'm developing. It's not like one product company. My company is a platform technology stuff. So I was able to always generate some money, keep me going. So I never had a problem. And I polyurethane technology, a license to Abrex, so I get a constant stream of um, licensing and royalty payments. That is helping me to develop new stuff.
Yeah, no, I think your business strategy is very interesting as well, which we'll cover a little bit later. But every time I talk to you raw, I feel like I'm getting a chemistry lesson, except I'm not a very good student, and I spend too little time on this. So I want to get some professor lecture from you about this whole beyond PLGA bioabsorbable polymer lecture. So you already talked a little bit about PLGA as the foundational
Right.
bioabsorbable polymer
Right.
that was discovered, like you said, a wonderful history of DuPont. But then there was a lot of problem with it. What was the problem with it for healthcare reasons?
Problem with the PLGA polymers?
Yes. Like without your polymers, what if we just use that? What's the problem?
What's the problem with the PLGA polymers? That's what I want. Okay.
Yeah.
PLGA polymers is the number one problem is very difficult to make it, very difficult to process them. PLGA polymers. And there's a lot of acid dumping when the hydrolyze too fast. So the acid dumping, which kills the cells, so it cannot be used for drug delivery polymer stuff. So having a copolymers reduce the acidity of the glycolide polymer. That's why caprolactone caprolactone is very very good polymer to work with. Is an excellent bio ink for 3D bioprinting stuff. Copolymers with the caprolactone easy to work with, and having a copolymer of a caprolactone in a polyurethane backbone is great asset for biocompatibility. Great asset biocompatibility. So over the years, after copolymers were done, because there was not a crying demand for new suture materials, Ethicon and Medtronic, they were not doing any new material stuff. So after 25 years, and my product was released in 1993, it's over 30 years, Medtronic just released a competition, just released a couple of weeks ago, competition for monocle suture. So all these years, there was no competition. Nobody is working on this stuff. That's one of the reasons, Jenny, that I wanted to develop other polymers from the knowledge I gained from commercial absorbable materials. I gained the knowledge of using these hydrolyzable links, which were in the absorbable polymer. So I take the link and put it in wherever I can, develop a novel class of polymers. I give you a simple example. Suppose you want to make a amino acid based polyester amides. You have a amine group, amino acids have amine group and carboxyl group. Glycolic acid, a hydroxyl group and carboxyl group. So I can functionalize them. That means I react them with an amino acid like a glycine and then make a polymer out of it. It'll become a polyester amides. So the glycolic acid link in the chain creates an absorbable polymer. I have over 15 patents on polyamino acid based polyester amides. So I can take a biostable polymer, I can convert into biodegradable polymer. I give you when I talk about absorbable polyurethanes, I can explain to you more. So I give you, before I go there, I want to show you we combined compiled polyester amides, polyoxyesters, and polyurethanes, and any polymer, like if you have an aromatic molecule, if a drug, let's say I have a drug which has a dihydroxy compound, dicarboxylic acid compound. Like anti-inflammatory drugs have all dicarboxyl groups and hydroxyl groups. I take that product and functionalize with the glycolic lactic acids or a caprolactone or PDO, and that's become a new entity and make a polymer out of it. So what happens, you incorporate the glycolic acid in the polymer backbone of the stuff. So you make an extrudable product or a drug delivery polymer, whichever, and in the presence of in the presence of moisture in the body, it starts breaking up the glycolic acid links. Okay, that's the same thing. I used it in polyurethanes, absorbable polyurethane. Nobody ever made an absorbable polyurethane except me. I'm the only one. If you go look Copilot, I'm the only, Bezwada Biomaterials is the only company have an absorbable polyurethane, controlled degradation profile polyurethanes. Use that technology for developing products like tissue bond and montage and all this stuff. If you have time, I can explain to you basic chemistry of polyurethanes. I don't think you have ever heard before. To you, can I
Yeah.
explain?
Yes. Also, I just want to add that we're going to have some graphic presentation of these compounds, so the listeners can go onto our YouTube channel to see the graphic demonstration of what Rod is about to say, because I understand the concept may be complex without any kind of visual explanation. So yes, go ahead and talk about polyurethane, because I know that is a main product from your company and your inventions.
Right. So, as I mentioned to you before, from the knowledge we gained from absorbable polymers, glycolic acid, lactic acid. So absorbable polymers degrade anywhere from 70 days to 3 years. So if I have a polylactide polymer, it takes 2 to 3 years for absorption. If I use a glycolic acid polymer, it goes to 90 to 100 days. Lactone goes to 91 to 100 days. So using that hydrolyzable links into the putting into the backbone of polyurethane creates a bioabsorbable polyurethanes. Let me go back and briefly explain polyurethane. Absorbable polyurethane is a polyurethane, biostable polyurethane is very useful, very unique, amazing polymer. But the problem with absorbable polyurethanes is it is the ingredients we put in, like toluene diisocyanate or MDI, which is a metaphenylene diisocyanate. Toluene diisocyanate is a carcinogenic.
I know.
MDI,
I heard cyanide. That's not good.
yeah, and the MDI is toxic. I mean, there's byproducts are toxic.
Right.
So no medical device use toluene diisocyanate. People use MDI, metaphenylene diisocyanate. It's a beautiful product, except that minor impurity of the toxicity. His rate of reaction is beautiful. Polyurethanes have a rate of reaction is very important. If it doesn't react fast enough, it's not going to be good for a lot of applications, like a tissue adhesives and the bone application products for bone application. So we took as the MDI is the key component for a biostable polyurethanes. My goal is to take a molecule very similar to MDI. MDI is actually two aromatic rings connected with the methylene group and diisocyanates on both sides. It's a simple molecule. The MDA, precursor of MDI, is toxic. So what my goal was to take that MDI molecule with the methylene group, replace that methylene group with a glycolic acid or lactic acid or caprolactone, so that resulting polyurethane becomes a controlled degradation profile, shorter, faster, and slow. So in compounds I use are very safe compounds to make that. I use aminophenol, which is a precursor of Tylenol, aminobenzoic acid, which is a precursor of sunscreen lotion. So you take these molecules, combined together with the glycolic lactic acid, which is the first step, literally a first step of the suture polymer, diethylene glycol with the glycolic acid on both sides. Link that into this MDI middle methyl, take the methylene group out and put that linker, hydrazable linker in the middle. So what happens is that molecule have a controlled degradation profile with no toxic materials coming out. That's how we convinced FDA that whatever put in is safe, and when they hydrolyze, they come out safe. So that's how we got 15 510K approvals. Abrex got 15 510K approvals. Matter of 18 months, they were able to develop a product. Now they have over 10 products for all orthopedic applications. So that is the key. So the polyurethanes are made with hard segment and soft segment. Hard segment contains MDI, soft segment contains polyesters. Whatever everybody doing absorbable polyurethanes is that hydrolysis of the polyester, soft segment. Nobody touches the hard segment of MDI. They have a partial hydrolysis, never get a full, complete hydrolysis. There, that's where I use this technology to make bio-based, biodegradable polyurethanes, where everything degrades to the starting material. That's what the new product we are trying to introduce to the.
I
So
have
that
a couple of layman questions about this
sure
polyurethane product. Is
good.
one, how do you know the bioproduct is going to be at the end of the degradation? How do you know? How do you measure it to see if there's something toxic to the cells?
Okay,
And,
okay. So what had happens is that if you look at the structure, you have if it goes down to aminobenzoic acid, so you got a worst can come only link of urethane, one link of urethane linkage in the chain. But they essentially what we did, Niti participated in making this stuff. What we did is that we synthesized the molecule and do the NMR interpretation
I see.
while we doing the stuff. So they break down to the starting materials.
Okay,
There's no
that's
byproduct coming.
that's clarifying. Yeah.
Then the major trouble is that the aromatic diamines are the cause cancer, not cancer, the toxicity coming from. But besides that, everything else is okay. That's why when they use MDI for the medical products, they extract the product out with the hexane solvent to remove all the impurities from there. So they will never use it as such. They make sure all the extractables coming out before they can use it.
And you mentioned that you have a partnership with Abrex and you had rapid FDA clearance. And I think our listener will be interested in knowing what these products are and what are the major application of this kind of polyurethane polymer.
Okay. So the Abrex is interested only for orthopedic applications, and I license the technology only for orthopedic applications. The main quality for these Abrex products are they're supposed to react very fast, become harden, because they use the calcium phosphate, hydroxyapatite. It's like a putty. So when you apply the putty, apply on the bone, it has to be spreadable. and also you can combine them as the adhesive, because it'll become, if I use only for a hemostat, it'll stay as a hemostat, react and becomes this. If I use it two bones to stick together within 45 seconds to 1 minute, they can start adhering together, become very hard like a bone-like material. So they have approvals for all kinds of applications: bone hemostat, bone cement, bone wide filler. Now in antibiotics, they have the antibiotic stuff, and have a thoracic surgery. It's a big hit on thoracic surgery, because when you cut it open, the ribs are
Yes.
bleeding. So when you apply this one before the surgery, and then remove after the surgery, remove it, and before they close it, you apply again this. It will gives, according to the literature, it gives about 30% strength, especially older people after surgery. My understanding is the big hit on thoracic surgery with this stuff.
Yeah, I see. So do they still use wires to? Because I read chest X-ray every day, and I see a lot of people have these wires on their sternum
Right.
because they had open chest. Do you put this putty on top of the wire, or do you still
No,
need the
no, no.
wire?
When you cut the ribs out, what do you call sternum in the middle?
Yeah,
uh,
sternum. Yeah, in the
Yeah,
middle.
I think that's where they use that,
I
and the
see.
rib, right? So they can take it out. Then when it is done, because it will prevent, um, the bleeding, and it also give us strength because it is already adhered to the stuff.
That's amazing. And have
Yeah.
they gotten any post-market analysis of bone regeneration or absorption for this kind of
Yeah, they are
putty?
publishing it. So my understanding is that because we are using a particular monomer stuff, you can control it. If for some applications you want to go slow absorption, so the bone can grow in with the stuff. It could go up to 2 years. As uh, the putty goes absorbable, then the bone will grow into the stuff. That is a neat thing about this Abrex
That's amazing.
technology. Yeah, yeah.
Yeah. I would love to know more about it, especially how the
It,
clinical outcome for these patients.
it's a great successful company.
So now we're talking about an external company that you work with. It seems like you work with many different companies through partnerships and licensing. How do people work with you as a partner? How do you find these partners?
So mostly I get the calls from somebody recommended somebody, or we talk to, the website, people see in the website. They will come and say, we have this issue um, with the product, and we will talk to them, and then Niti will be there as a quality person with the stuff, talk to them what the issue is, and then we go back and make a prototype of the sample, and after that we go and explain the stuff to them, uh, and make the samples, prototype samples, and they evaluate it. They come back and say, we this is good, but this is not good. The strength is good, but absorption is too fast. So you fix this information. If we go back again, do the stuff, correct the points, and go. And then once they approve it, the sample will have a scale up capacity, have ISO 9001, 13485 approval stuff. As a team, we look into this and we'll work on the project stuff. That's how.
So
Um,
not only do you design the polymers, you also help them with manufacturing. Is that right?
oh, yes, 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 yes. We do. Um, well, we are working with a customer now on the electrostatic spinning tissue scaffold stuff. So
Yeah.
they look very promising with the stuff we have done. We have done with the small scale in university tissue scaffold stuff. Then we have a commercial customer came to us to work on tissue in scaffold, and that's what we're doing. We submitted two polymers for tissue scaffolding, And they came back and said electrospinning problem. And they said one is good, one is not good. We got to talk to them and find out what the difference is about. So it's a constant communication back and forth. We with the Zoom calls and presentation stuff like that.
Yeah, but it's a universal. A lot of people don't know that's happening in the background. We met each other many years ago, probably starting in 2020. Is that right, Raul? Was it
Right,
2020?
right,
And our journeys intersected because of 3D printing and bioprinting.
right,
And I
right,
know you have a very strong interest in this field as well.
right, right.
What is different about bioabsorbable polymers for biomanufacturing in particular? You mentioned electrospinning, which is very interesting, by the way, because we just had a podcast with Professor Paul Dalton, who is the inventor of melt electrowriting, which is
Mm,
like a modified version of it's like a more controlled way of 3D printing using electro spinning foundational information. So I'm just curious, like, how did you become interested in 3D printing? And what are your
very,
take on polymers in bioprinting and biofabrication?
yeah, very interesting question. It's a very good question too. So our participation in 3D printing is accidental. So what happened? A friend of mine who was working on 3D printing about 2000. Um, they're interested in peg acrylates. So we have no experience on making peg acrylates. We know how to make it, but he never made it. So my friend came and said, I need it bad. I need large quantities of the peg acrylates. I have nobody to make the product for me. You got to help me out. So I end up making for her. We was happy with the stuff. They're very happy. And we work with a big customer, a very big customer, on making over 200 kilos peg acrylates with 6,000, 3,400, all kinds of peg acrylates. And they were very happy with our service and they wanted us to make other peg acrylates, methacrylates, peg all this stuff. So that is how we got into this peg business stuff. So
One might be
now,
curious. One might be curious what they're making that they need so much.
yeah,
If
yeah,
if you're allowed
we
to
don't
tell
know.
us.
We don't know. It's a big company. We don't know what they're
Okay.
making with this stuff. So we found out there's a lot of need for this stuff. So we have a whole new peg polymers, and we work with the two or three small companies who are working on breast implants, organ replacement stuff. So we work with them, and they say peg is weak. I don't have elasticity, no strength. Can you help me out? So we develop new polymers, excluding glycolide. Glycolide is a bad actor with the peg copolymers. So we go with polycaprolactone, PEG copolymers, caprolactone, trimethylene carbonate, lactide. So we buy diblock copolymer, triglock copolymers, gives enough strength with this stuff. Some of the customers, we know they are working on breast implants to improve the strength and the stuff like that. That's how we got into. Now we are almost equivalent to some of the peg suppliers. We have more peg polymers than some of the supplier stuff. We are getting only not for any other medical pharmaceutical product. We're getting only for 3D bioprinting work. Our peg polymers we're making with this stuff right now. And I would say we have at least 50 to 80 peg-based polymers in stock. We are supplying companies like the Sigma Rich and distributor stuff, but we are working on also small companies who needs this product stuff. And if you know anyone wants to peg acrylates with a good price, we are here.
What do you think is the challenging when you were developing these polymers specifically for bioprinting applications?
So I could give you an example of pegs. Peg is one example, and I give you another example to PLGA polymer. Peg polymers, we really mastered peg polymers. Actually, we're going to have a couple of patents on incorporation of the polyurethane to the peg polymers to give a extraordinary strength. And also, we have discovered that you can make sealants, peg-based sealants with the beautiful strength, adhesion, all this stuff. So we talk to the customer and we see what is happening, and we develop based on the customer say, now I am working on this. I have a problem of solubilizing it. How could I make a diblock copolymer with a good solubility in water? Polyoxyesters is another example. Polyoxyesters containing glycolic acid moieties in the stuff, but it behaves like a peg, but it have polyester strength. So I can make, if somebody wants only peg, nothing else, with the strength with the stuff. So I can use polyoxyesters and the backbone of the chain, and then we have pendant hydroxyl groups and convert them into acrylate groups and photocurable with this stuff. So I have a polyester backbone with hydrolyzable links like pegs, and have a pendant groups with the stuff, and the cross link, either cross link with the peg acrylate groups, or attach, add the reactive isocyanate, hydrolyzable reactive isocyanate into this stuff. So you can cure them while you are at room temperature. You can cure them with pegylation, no, sorry, bioinks, bioprinting. That's where our direction is going with this stuff. So other example I want to give you quickly in PLGA. Customer is looking for a polymer, have a specific requirements, solubility, viscosity, hydrolysis, and coating properties of one polymer. So we're working back and forth with the customer. Is a big company customer. Develop one piece at a time. Work with the customer back and forth. It's going on very well. That's why we work with the coordination with the customers. Develop products. A lot of time, the customer have an issue, we're able to solve them. That's how we build the relationships.
Thank you so much. I learned so much from this episode. Now, we're reaching the end of this conversation, although I do hope it can last longer. So I want to hear some of your vision for the future. Where do you think Bezwada Technologies, Bezwada Biomedical is going to play a role in the future of healthcare and drug delivery, biofabrication, and maybe even sustainability, which is one topic we haven't really focused on. So just kind of want to see, where do you see your company is going to do to change the landscape for the industries?
I to be honest with you, Jenny, I thought it would have happened 5 years ago. You could have been a completely different company, but it's very difficult to get the funding, and very difficult for the companies to aggressively go after. Our tissue adhesive, we have met all the requirements for the tissue adhesive properties, and it is safe and biocompatible and good tensile good properties. But the funding is the difficult one. But we are struggling to get more funding to develop the stuff. What I see is I see a lot of opportunity 3D bioprinting, even though it's a little bit slower right now. We feel the technology we have, especially with the polyurethane into the system, is so we can make tissue at room temperature without heating, no heating, with very quick reaction stuff, biocompatible polymers. That's for 3D bioprinting. And I also see biodegradable polyurethanes. We have tested it. Which is biodegradable in the soil uh, testing. We proved that we can make a biodegradable, uh, bio-based polyurethanes. We have done extensive work, but there is no big money in there. But I'm committed to do at least, if I reduce the polyurethane waste from 25 million tons of polyurethane waste, even if I reduce 1 ton or 2 tons of the polyurethane waste, I think I make it there for me. It's going to be fantastic. That's one. And I'm going to, other thing I'm struggling for years. I thought this is going to be a great thing is that bioconjugated drugs. So my technology have so many patents and biotechnology, drugs containing hydroxyl group, carboxyl group, amine group. I can react them, make a polymer chain, and have this drug in the chain, and then you want to trigger by in the moisture, in the presence of moisture, breaks down in a controlled fashion. If I use the glycolic acid, it'll go very fast. If I use lactic acid, it goes slow. Caprolactone, very slow. I can control the degradation profile. I'm working very hard now trying to get a funding. It's so difficult. I've been trying actually for many years and nobody wants to hear it. Everybody wants a ready-made product. They want to take it and sell it. Nobody wants to invest time to develop the technology. I think that's one of the um, unfortunate thing I have for my business, not able to get funding together with this stuff. But I feel that with 3D bioprinting, I have a lot of hopes on that.
Well, I certainly do as well, since I'm heavily invested in this industry. And I hear you, Ra, and I have to say your perspectives are very unique and it's definitely a chemist perspective. You know, nobody else is going to measure their legacy with a ton of polyurethane waste as a legacy metrics. You know,
Right.
you're the only one I've ever heard of.
I wrote
Yeah.
to all the big companies. Nobody responded. Everybody thinks the cost is a problem. They don't want to touch it because now the regulations are really relaxed on the polymer waste. Um, so it is difficult.
Yeah. I understand where you're coming from. A lot of things depends on what you work in, day out, what you're seeing, the world that you're in. Like our perspectives are different. And I actually recently wrote an article about why are we spending so much money in AI and
Mm.
not enough in
Right, right,
biofabrication and
right,
science, basically. And
right.
you know, it's a much deeper question that I think you and I constantly are thinking about this. And it's a combination of culture, policies,
Right.
politics, and humanity. What do we prioritize? And unfortunately, at the moment, I feel science has taken a step back
Yeah.
and a lot of other things are more prioritized.
Right.
You know, I don't know. I mean, for me, for you, this probably is a bit of misjudgment, but for many other people, AI is probably the number one.
Right.
And I'm not saying I'm right. I just don't know. I think time will tell. Um, And I am also convinced that we need to invest more in material science, in biotech in general. So yeah, I'm on the same page, but I understand your frustration as well. Now, for the final question I have for you, what I love about you is you have this entrepreneur spirit all the time. I mean, you have 40 years in this and you're still as enthusiastic as you were 40 years ago. I don't think that this has diminished over time. What are your some advice for the next generation of scientists and entrepreneurs in medtech in particular? What do you have in terms of advice for them?
I think don't lose hope on the science, even though it took a back seat now. Um, I have a couple of uh, uh, co-op students work with me. My advice to them is to have a deep understanding. I think what happening now, everybody come in, they want to learn quick some things and get a promotions, move on. It is not unlike old days. Nobody is interested why is working, why is not working, what makes it product successful, not successful. Um, my advice is to learn, understand what you're doing, have a logical thinking, because most of the people work now, college kids, they don't care about this. They come in and go home and work on other stuff more than the work. That's what I noticed from biomedical engineering students worked with me, co-op students, master's graduates with this stuff.
Well, we're definitely a lot more distractible these days. You are a focused person, that's for sure.
Yeah.
Thank you so much, Rob. This is a wonderful conversation. I hope to see you again.
Okay.
Talk soon.
Thank you.
Okay.
Yeah. Bye.
This podcast is for educational and informational purposes only. The views expressed do not constitute medical or financial advice. The technologies and procedures discussed may not be commercially available or suitable for every case. Always consult with a licensed professional.